Okay, let's continue with the program. This will be the last topic section. It's entitled Microbiome and Health. Really is an important topic. All these studies in microbiome, eventually the idea is that have practical implication, especially in the clinical part. So the outline of this session will be, first I will give a briefly introduction about the topic, very essentially to try to confirm and to reinforce the central ideas that has been exposed here with different topics related with microbiome and diseases. In particular, in this candidate in the Computational and Systems Biology program at MIT Association. Make hypotheses, testable hypotheses, in order to understand what is occurring inside one microorganism. In order to do that, our strategy is the following. We will follow a paradigm in systems biology that essentially consists, once we have defined the organism of interest, for instance, E. coli, or a human cells, or Rhizobium etli, essentially we use high throughput technology like transcriptome, genome, proteome, Jorge Vázquez sense in a mathematical components. Essentially, this matrix contains information about the rows represent the metabolites, and the columns represent the reaction, the transformation, the chemical transformation that can occur inside the system. Currently, this paradigm, systems biology, has been applied for a variety of organisms that includes archaea, eukaryote, and bacteria. This is one more specific information, don't seem too much the question, but all this genome information that came from this reconstruction can be included in this matrix, and we can show that this equation can be applied relating the metabolic reaction that occurs in one organism, the thermodynamics flux, the metabolic flux associated for every one of the reactions that you include in the reconstruction, and this represents the temporal change of the metabolites. So it's a very simple link to relate the genome information with the possibility to model in a quantitative way. With this simple equation, we can explore the metabolic capacities that one specific organism can be able to produce. Even we can explore and you can make hypotheses of which one are the central metabolic reactions that has an important influence to define the phenotype behavior. This is the global sense. For instance, this is a clear example. The systems biology of microorganisms, this is the reconstruction of Rhizobium etli. It's a bacterium that fixes nitrogen. And in 2007, we published this paper in collaboration with Bernard Palsson at UCSD. Essentially, this is the reconstruction, contains almost 500 metabolic reactions for this specific organism, Rhizobium etli. And with this set of metabolic reactions, we introduce all this information in a computer, and then we predict certain phenotype. In this case, we were interested to modeling the nitrogen fixation. But you can model in the growth rate, the biomass production, for instance, another type of phenotypes. What combined this type of prediction in silico with high throughput data? In this case, for this organism, we combine transcriptome data, proteome data, and metabolome data, all together in combination with this computational model. The predictions that you can obtain are extremely interesting because you can say, for instance, there are certain metabolic reaction in all this network that does not change, does not have any effect in the phenotype. In this case, the nitrogen fixation. So you can prove, you can test, you can suggest hypotheses. Applying this simple logic, the next point, our objective, is to try, for the case of the microbiome, is to try to make, once you have the sample for one, let's say, stool, human stool, for instance, our objective is to try to define a reduced community of organisms in terms of functionality, maybe, and to represent every one of these organisms through a metabolic reconstruction. Once we have the input in terms of the metabolite that there is in the environment, it's possible to make the simulation and eventually to predict, to suggest which one will be the metabolites that represent the output of this complex system. You can make a variety of hypotheses. For instance, delete one of the organisms, and what will happen with the output in terms of the metabolism. And the central point, our challenge, is to try to this output metabolism in terms of the phenotype, in this case for the human. So we are very interested to impose systems biology in terms of this type of formalism of paradigms used to study human diseases. We will have one meeting from 2 to 4 of August next, and it's involved, of course, the Center for Complexity here in UNAM. Finally, I want to say the people that has been involved in this type of efforts is UNAM, Wright, and the people is my team and the people from Wright College. So let's continue with the part from the speakers. The first speaker will be Shirley. Okay, go ahead.
Thank you. Thank you very much for inviting me to this symposium. I will talk you about studying the gut biome in Mexican obese children. Childhood obesity is defined as the body mass index at or above 95th percentile for children or same age and sex. And is defined also that excessive accumulation of fat. Lamentably, Mexico ranks the first place in the world, and prevalence in children between 5 to 12 years old is 15%. The prevalence of childhood obesity is increasing in developed and underdeveloped countries. Obesity is a multifactorial disease that is caused by the interaction of different factors that, like environmental, genetics, metabolism, social, and cultural factors. And the obesity constitutes a risk factor of obesity in adulthood and a potential risk factor to development chronic disease such cardiovascular disease and diabetes. Now, in recent years, the gut microbiome was associated with the development of obesity. It has seen that there is changes in the proportion of firmicutes and bacteroids in obese children or obese individuals with respect to healthy individuals. And there was an interesting study where transplant microbiota from obese twin to a germ-free mice perform an obesity phenotype in the mice. And the transplant of microbiota from lean twin do not make the obesity phenotype. But in most recent years, the gut biome was associated with changes in the gut microbiome. The gut biome have the majority proportion of viruses in the gut are bacteriophagous. Bacteriophagous regulates the bacterial composition and functions of their host. And dysbiosis in the gut microbiome could be association with the function of these bacteriophagous. There are very few studies about the relation between phages and their host bacteria, but there are an interesting study where in the intestinal bowel disease analysis, they found it that an increasing in caudaviral phase was associated with a decrease in bacteroides and increase with Enterobacterias, Pasteurella, and Prevotella. This bacteria was associated with this disease. Another interesting study was they analyzed the viruses that are transferred through the fecal microbiota transfer to treat ulcerative colitis. And an interesting point was that they saw that the donor had a viral families different. Some of the viral families were different to the patients with ulcerative colitis. When the transplant was performed, the patients 15 weeks post-transplant, patients maintained, in this case, 5 viral contigs of the donor, 7 viral contigs of the donor, and in patient 3, 34 donor viral contigs. And they saw that the preferential transfer was of temperate bacteriophagous. It suggested that the fecal microbiota transfer could improve the ulcerative colitis not only by the bacteria that are transferred, but viruses could be an important role in this transfer processing. So we want to know what happened in obesity with the gut virome. For this, we analyzed the fecal samples from 3 healthy children and 3 obese children between 7 and 10 years old. And we isolated the viral particles of the samples. We verified that we obtained the viral particles by electronic microscope, and we performed the metagenomic sequencing of the viral particles. Of the total risk that we obtained, only between 4 and 8% of the risk mapped to human. This is an important data because many studies have mapped to 90% of human data. So we analyzed the taxonomic distribution of female viruses, and we could see that there was different patterns between obese children and healthy children. And if we analyze the relative frequency, we saw that the Meioviridae family have a significant difference between healthy and obese children. And Cyphobaridae have a significant correlation too. But to understand better the role of viruses in the gut microbiome, we analyzed also the 16S taxonomic classification. We saw that the pattern of healthy children and the pattern of obese children at phylum level, and we saw the pattern of healthy and obese children at genus level. We could see that Firmicutes have a relative abundance, a major relative abundance with respect to healthy, the majority of healthy patients. At genus level, Prevotella was abundance in healthy patients, in difference with obese children, where Blautia and Bifidobacterium and Lactobacillus and Bacteroides were the predominant genus. So we analyzed the association between the viruses and bacteria that are in the gut microbiome of these children. At phylum level, we could see that actinobacteria, that the Inoviridae family do not correlation with the relative abundance of actinobacteria. However, if we see the relative abundance of Inoviridae with actinobacteria, there was an important correlation in relative abundance. We saw also that Inoviridae don't have a correlation with the relative abundance with Bacteroidetes, but in obese children, they have an important correlation too. Inoviridae with Firmicutes do not have a correlation in healthy children, but they have an important negative correlation. The increasing in Inoviridae was associated with decrease with Firmicutes. At genus level, Siphoviridae was associated, the increase of Siphoviridae was associated with an increase with Bacteroides in healthy children, and have a negative correlation in obese children. Siphoviridae with Bifidobacteria have a positive correlation in healthy children. However, in obese children, there was not correlation. And in with respect to Bifidobacteria, Inoviridae have not a good correlation, but in obese children, they have a positive correlation. This data suggests that in obese children seems to be differences in relative abundance between healthy and obese children. And the presence of some viruses could be associated with changes of some bacteria that I present in these graphs. We need to analyze more sample to get a better analysis of obeses, and it's important not only for a better understanding in obesity, the viromics data we will integrate with metagenomic data and metatranscriptomics data. They are areas that are investigated in our laboratory to have an integrated idea of the role of gut microbiome in the obese children. Thank you for the microbiomics labs and in Mehen. And sorry for my English.
Thank you, Shirley. Let's move to the next speaker, Jorge Vázquez.
Well, so good morning, everyone. Today, I would like to, also thanks for the invitation. Today, I'm going to talk about this, the functional gut microbiota and its association with chronic immune activation in the HIV infection. This work have been performed in the University of Valencia and in la Fundación para el Fomento de la Investigación Sanitaria y Medicina de la Comunidad Valenciana. So maybe as you know, HIV is a global pandemic which actually affecting almost 37 million people, from which fortunately almost 16 million people have access to this antiretroviral therapy. And the use of GARD have increased considerably the life expectancy by restoring the CD4 T cell count and maintaining a really low level of the viral load. Indeed, there is no risk of infect your sexual partner, even if you are not using any protection or condoms. So, however, the life expectancy of this group of patients is 10 years lower than the average of the population. And currently, this reduction is given to unrelated illness, such as cardiovascular diseases and atherosclerosis, that are caused by persistent immune activation and chronic inflammation. And this is because, this could be explained because this chronic inflammation could be explained by the constant bacterial translocation from the intestinal lumen to the systemic circulation, giving a prior disruption of the gut-associated lymph tissue. So this is happening because at the early stages of the HIV infection, the virus deplete the Th70 CD4 T cells, which are, and also the loss of these cells and the infection itself cause a lot of abnormalities into the intestinal lumen, such as abnormal enterocyte differentiation, enterocyte apoptosis, the loss of the type function, decrease into the IgA production, and the loss of the capacity for the recruitment of neutrophils and macrophages. Once this revolution happens, then the microbiota starts the translocation of several microbial compounds, such as peptidoglycan, lipoprotein acid, lipopolysaccharide, flagynyl, ribosomal DNA, or methyl CpG containing DNA. And this is not just, accompanying with this disruption, there is also, in 2013, Lo Suppone et al. Showed that actually exists a dysbiosis given this HIV infection, in which these HIV-infected patients' gut microbiota associated is different from the one that's the controls in the US population. And also, the chronic ones, the patients that have been living with more time with the virus, resembles a microbiota that is closer with these Amerindian communities. It's known for have a agrarian diet, are rich and are also rich in prototype species. Also, these have been improved other alternate sequence technology and other fecal and anal swabs have been improved by several approximations. And Bukovic et al. Also showed this dysbiosis, but more importantly, that exists a correlation between the dysbiosis and the T cell activation. So, So there are a lot of questions about this HIV gut-associated dysbiosis, but today I would like to talk about the characterization of gene content and taxonomic composition of different HIV group of patients, and the use of the probiotic treatment to restore the microbial gut-associated community. So in order to achieve this goal, and in collaboration with Hospital Ramón y Cajal from Madrid, we recruit a cohort for 44 patients, from which are 15 controls, 17 patients with good response to the treatment, 10 patients with no response to the treatment, and 11 naive ones, HIV positive naive ones. They are naive, they haven't started treatment because the CD4 T cell counts are okay and their viral loads are quite low. And we apply a 6 weeks intervention for a mixture of 20 grams of probiotic of short-chain galactoligosaccharide and long-chain fructooligosaccharides plus glutamine to restore the mucin. and also a placebo group. And after the 6-week intervention, we managed to 44 patients finish this intervention. The exclusion criteria was the use of concomitant medications, the use of systemic antibiotics during the previous 3 months, other comorbidities or GI symptoms, and in collaboration with also the Centro Sandoval y el Hospital Gregorio Marañón, also from Madrid, we took plasma biomarkers related to inflammation, thrombosis, bacterial translocation, endothelial function, also the thymic function, and the T-cell marker from the CD4 and the CD8 T-cell count, as well as several metabolic profiles in plasma. Well, also, we measure a lot of genes of expression related to the HIV immunopathogens, but today I don't want to talk about it. The metagenomic workflow was quite easy. Just took the stool samples, and then we obtained this is the cake from one of other really for the PhD for the PhD celebration from one of other the PhD students in the lab. And then, well, we just take the bacterial suspension and we extract the DNA from the KIAM kit by this column of affinity columns and amplify the 16 ribosomal RNA and parasequence it. And then we're just analyzing with the state-of-the-art pipelines, such as CHIME and R or LEPSE. In a similar manner, we just took all the whole genome and it went then for shotgun sequencing, and a combination of parasequencing and Illumina V3 sequencing, we sequence all the whole microbial shotgun DNA. And again, we just apply a established pipeline in which just stream adapter, we remove sequence artifact filters. We just perform a quality control by just trimming the bad quality sequences. We just clean for all human possible host contamination, and we assemble using 2 different strategies. Then, the assemblies were then used for predict-ORF, they were closed using uClust, and then we use RapSearch, the cyst of the uClust, when they're used to be launched with RapSearch to different databases. Also, with the unassembled reads, we just map, again, all the genomes available in the RepSec and in the Human Microbiome Consortia database. So our first question was, does HIV cause changes in the bacterial composition of the microbiota? And the answer is yes. So we observed 2 main clusters. In blue, one cluster that contains most of HIV uninfected individuals, and in red, a cluster that contains most of the HIV-infected individuals. And this difference was highlighted when we performed the pairwise comparison between the 3 different HIV-infected groups against the controls, and we observed that there is still a statistical difference into this microbial composition. And it's also interesting that even the good response to the treatment, to the antiretroviral therapy, is not enough to restore the good microbial composition. So we were asking which are those species that were overrepresented, and when we observed that actually when we obtain the gram-negative, gram-positive ratio, there is 4 times bigger in the HIV-infected patient than the one observed in the controls. And this enrichment is mainly given by the overgrowth of the Prevotella succiniviro and Aspergillococcus species, and a deplete of species from the Bacteroides, and more important, from Faecalibacterium. The Faecalibacterium parotidis, this species is really important because this stimulates T-cell regulatories, the TREC-cell, CD4, CD8, alpha-alpha, which are really important to the immunosuppression by the secretion of the interleukin-10. So also we measure the diversity and region estimator using 4 different indexes, and what we observe is actually the use of the art decrease the diversity into these gut-associated communities. But besides of the use of this of the antiretroviral therapy, we also ask which other variables could be related to the decrease of the diversity. And what we observe is that actually the time that these patients have been living with the infection, and these 2 markers that are related with the damage that the virus have done over the mucosa, the maximal viral load that the patient have ever had, or the minimal number of CD4 T-cell counts, this tenadir, have a significant correlation with the decrease of the diversity, which implies that maybe as much time have you living with the virus, and as much the damage that the virus have performed over the mucosa, then the loss of the diversity into this microbial composition, microbiotas. Our second question was, does HIV cause changes in the gene composition of bacterial good microbiota? And again, yes. We observe also 2 groups, and the pairwise difference are again statistically significant, also the cake orthology database. And also, again, even the good response to the treatment is not enough to restore the gut-associated functional community. So then we ask which are those genes, those pathways that are represented in the HIV gut-associated communities. And what we observe in red is that actual increase of pathways related to the infection diseases, pathways related to the energy metabolism, pathways related to the synthesis of amino acids, and some pathways related to the terpenoid metabolism. And as well, there is a decrease of pathways related to the lipid metabolism, carbohydrate metabolism, and some vitamin metabolism, and signal transduction and Weymann transport. But more importantly, and is congruent with the enrichment of the gram-negative community, we observe that actually there is an increase of genes related to the lipopolysaccharide biosynthesis, which is an important component of these gram-negative bacteria. But more importantly, this is a metabolite that triggers the innate immune response by inflammation, and it is really fast recognized by the monocytes. So our third question was, is there a connection between dysbiotic microbiota, bacterial translocation, and immune activation? By doing this, we model it using a Bayesian network. Just Bayesian network adjusts probabilistic graph, directed probabilistic graph, in which nodes correspond random variables, and the arcs correspond to conditional dependencies. These Bayesian networks have a property which is called the macro blanket, which is the set of nodes that renders it independent from the rest of the network, for the rest of the network. So what actually we're looking is just dissect the network for important metabolites or pathways that could explain, that will be related to the systemic immune activation. So we perform the Bayesian network using the KET pathways, the genus, and the T cell activation, and the variables for T cell activation, thymic function, and bacterial translocation. So what we observe when we just subtract the lipopolysaccharide biosynthesis Markov blanket, we observe that actually have dependencies with, as expected, with some gram-negative species, some pathogenic pathways, and pathways related with the resistance of the oxidative stress. And also, I think really important, have direct associations with variables related to the immune response and with the PPI, which is a marker for the bacterial permeability. So all of this together suggests a scenario in which a pathogenic gram-negative enriched community adapted to the oxidative stress of this inflammatory mucosa is promoting systemic immune response by this by the translocation of these bacterial metabolites. So finally, we asked, do a probiotic could help? And short answer, no, because we couldn't separate between the basal condition, the placebo, or the probiotic, even with the genes, the orthologs, or the O2s. But when we perform this analysis, just focuses in the response of each group. In the case of the HIV naive group, the HIV ART naive, we perform a clustering analysis using the basal condition of the controls and the three groups, the basal condition, the probiotic, and the placebo of this group of the HIV positive naive ART. And what we observed is a lot of translocation between samples of these two clusters, which indicate that there are some characteristics that are in common and could be susceptible for the placebo, for the probiotic administration. And when we perform the LEPS analysis, which is we observed that there is an increase of, using the basal condition versus the probiotic, we observed that there is an increase of bacteria related to butyrate production, also genes related to the butyrate production. And just remember that the butyrate is really important because this compound promotes reduce the systemic the local inflammation of the gut, and is the main source of energy of the enterocytes. Almost 70% of the energy of the enterocytes are given by the butyrate acid. Also, we found other pathways related to the short chain fatty production and species related to reduce the hydrogen, which is an element that poisons and stops the production of the butyrate acid. But also, we also found species that attenuate immune response, such as the Faecalibacterium and some bifidobacteria. So, okay, so there is a microbiota that this probiotic administration maybe is enhancing the production of the butyric acid. So we just measure the production of butyric acid into this bacterial suspension, and we observe it actually just for the person treated is the only group that increased the butyric acid concentration after the prebiotic intervention. And again, we perform the Markov blanket of the butyric acid, and we observe is actually there is direct relationship with bacteria known for the production of butyric acid, and negative relation with markers related to bacterial translocations such as CD14 and with systemic immune inflammation. So, as a conclusion, also we observe this severe breakdown of the gut that allows the overgrowth of this altered microbiota enriched in gram-negative bacteria with this dysbiotic metabolic profile that is correlating with immune activation, even with the good response to the art, and also with this bacterial translocation. And the use of the probiotic, this nutritional intervention, is really dependent of the time and the health that have done over the mucosas. So maybe this probiotic is useful as soon as possible. Because maybe the virus haven't had enough time to disrupt the gut completely. So, and with the disruption of the gut completely, there is the dysbiosis, and with dysbiosis, there is the loss of the bacteria that are capable for the butyrate production. So this work, and it can be found in these two publications. Well, and this we are just waiting for the ISME reviewer to just finally accept the second publication. And also, we have done the metabolomic and the proteomic in this cohort of patients. And these days, we are actually working with the metatranscriptomic in order to have a holistic view of the whole HIV gut-associated dysbiosis and this response to the probiotic. And also, there are also papers related to bioinformatics and viral metagenomics and some of antibiotic resistance. So thank you for your attention. I would like to thank CONACYT and CECITY.
Thank you, Jorge. The next speaker is Mariana Matus. She is from MIT. Go ahead.
Thank you. Is this? Well, I will use this time to thank the organizers for the invitation. It's great to be here and to be knowing like all the people doing microbiome work here in Mexico. So in my talk, I will talk a bit more on how we want to use this microbiome work in order to create new diagnostics that are clinically relevant. And basically, our approach, our philosophy is that even before we know exactly what are the mechanisms behind the microbiome influencing our health, we may be able to use it to either better understand and diagnose certain diseases or to even treat it, as FMT has shown for Clostridium difficile infection. And what motivates us is to think about, if you think about getting diagnosed for a disease, you will immediately think about, going to a doctor, probably giving a blood sample that will go through, very well-established tests. There are several hundred blood tests already out there. But what we think is that actually, every day while you are at home, you are generating a bunch of health data that you're basically just flushing down the toilet. And our idea, and then if you look, if you go online and search for how many clinical tests are there for stool, there are like less than 20. So we are very motivated to really move forward the use of stool and wastewater as a way to learn about human health. During this talk, I will talk about two projects in particular. The first one is just starting in collaboration here with Osvaldo Reséndiz and other researchers. And the second one is one that we have been doing over at MIT in collaboration with Kuwait. And that has to do with using wastewater to learn about the health of communities. So in the case of personal health, I have several projects that, in which we have cohorts of chronic disease patients, and we're trying to better understand what is going to be their outcome in the long term. One of them that it just basically started early this year is, the team is over here. Oh, wait. Here's a team when they visited MIT back in March And as you can see, we have people from INMEGEN, the Red de Apoyo a la Investigación, Universidad de Guanajuato, MIT, and we have, besides having seed funds to do 16S sequencing of about 500 samples collected in Guanajuato, we also were fortunate enough to get support from the MIT Mexico initiative to fund travel between both countries over the next year and a half. So this was our kickstarting the project with this workshop at MIT. And basically, if you are aware of, if you're in touch with the clinical microbiome literature, you would know that there's already 3 big papers out there describing how the microbiome changes in type 2 diabetes as compared to healthy. This has been done in a Chinese cohort, in a Scandinavian cohort, and lately also in a Dutch cohort. So we're not that interested in describing that, which has already been done, but rather to focus our attention into pre-diabetic subjects, which are pretty heterogeneous, but most importantly, when you are pre-diabetic, you can still reverse to being like healthy again. Once you have completely gone to be type diabetic, there's nothing you can do. You have to take medication for life. So, as I mentioned before, our clinical collaborator at Guanajuato has a clinic in which he offers free screens for type 2 diabetes to any person who wants to get it. And he runs a lot of clinical assays and characterizations on them. He collects a stool and sero sample from every one of them. So what he has right now is already a few hundred samples, mostly from people that have different types of risks or already insulin problems and deficiencies in their metabolism, but not quite yet the disease. And we're going to be looking at that cohort first in understanding how can we better stratify this patient population and assess the risk of progressing into type 2 diabetes. We're going to be doing basically 16S analysis and correlation with all the clinical characterization that our collaborator has. But we also are interested in following a few of these patients longitudinally over the next few months and over the next few years in order to understand, in order to find biomarkers of disease progression and eventually understand what's the role of the microbiome in that. It's also very important that some of these people who come to his clinic are newly diagnosed type 2 diabetes. So I don't know if you know the statistics, but basically about 50% of the people who have type 2 diabetes in Mexico don't know they have it because the symptoms are, fatigue, and other not very clear symptoms. So many of the people who come in actually get diagnosed by him and are naive to any treatment. So he's allocating them into different types of therapies, and we are going to also be following their response. Next, I will talk about the work we've been doing at MIT to understand how can we use wastewater to learn about the health of a community. And here, our rationale for that is that, wastewater networks are very special because it's the one place in which we all are contributing every day our samples. So they naturally are aggregating all this health data. And many studies have been done in understanding how wastewater is treated and the quality of it going back to the environment. Our interest is to look at it as fresh as possible. So our project is very innovative. It's the first of its kind. And our idea is to have based on the tributary map, which is the wastewater map of a city, which is the mirror of the location, of the spatial configuration of a city, as well as the temporal dynamics of our behavior. We want to be able to go from a map like this into a platform that can be used to do public health. As an example for how this can be used, I like to talk about this. This work has already been done since the 1980s. It's not new. But it's fascinating, and I hope everybody would know about it. Like poliovirus, it's by now mostly contained and eradicated from most countries. But for a long time, but it's still endemic in a few. And for a long time, the only way to know that you have polio in your country or in your city was by getting kids who come to the doctor and they already have chronic paralysis problems, right? So the surveillance of the disease was based on kids getting sick and kids coming to the clinic with paralysis. But this is a very bad idea. This sucks. Because actually, like, only about less than 5% of the kids, no, less than 1% of the kids who have polio develop the symptoms. So by the moment that you receive a kid into your clinic and you diagnose this kid has polio, there are on average 99 others infected in your population. You don't know who they are, and they are just spreading the disease. In contrast to that, 96% of children who are infected will shed the virus in abundance in their feces. Therefore, there has been work, for example, this example in Israel, where even if they have been polio-free since 2002, they continued monitoring their wastewater for the virus. Very simple, just get some wastewater, do PCR, looking for the virus, and back in 2013, they have a big scare because they found the virus, the wild version of the virus again for the first time in like 11 years. And based on that, their Minister of Health allocated money to buy enough vaccines to cover all children in the affected areas. And this was really awesome because they actually, by the end of the year, they contained the virus and there was no paralysis cases whatsoever. The same happened in Egypt, and then by comparing the genetics of the viruses, they saw actually it was the same virus that had come from Nigeria into their countries. What we imagine is taking this, inspired by this work, we want to take this one step further and not only look for, in this case, polio, which in most countries is not a problem anymore, but what else can we learn about this? We imagine learning about not only other pathogens, but also the spread and the prevalence of antibiotic resistance, for which we have very little data right now to compile trends and statistics that are meaningful. We also imagine it as a way to monitor, for example, diet or lifestyle of communities, of cities, of countries, and tools to build analytics that one day can almost be fed in real time or semi-real time to inform public health authorities, but also to be used by researchers to understand at the population level what's happening. So this is a bit of the work we've been doing. As I was saying, sampling at a manhole is not very common. Usually researchers who work with wastewater will go to a treatment plant. So this is just to show you how we have actually had to have engineers in our team to help us develop technology to better sample. We started by having help from an officer from Public Works and just literally dipping a pole into the water and giving it back to us, which was really, really gross, as you can imagine, to going into buying a commercial peristaltic pump with which we could deposit water directly into a bottle without having to have water splashing everywhere, and that was great. And then, but also now we have our own robot that can, it goes down to the water level. It can collect water into the bottle, whatever volume you want, over the period of time that you want. But also, in the middle, we can plug in whatever we want. So we already tested plugging in a filter that will capture bacteria from the water. And that way, instead of bringing back bottles and bottles of water, you can bring already, filters with your bacteria. So our very first experiment was okay, so we're sampling at a manhole, and you can see there, this is the city of Cambridge. And over here, you can see in red the neighborhood that we sampled, and in black, the manhole at which we sampled, that represents that population. And that represents only about 2,500 people. And by then, the water is still very fresh. We calculate that the farthest point in our catchment area is only about 15 minutes away from where it's produced to where we collect it. And we contrasted that to samples that we collected at the treatment plant in Boston, or very near the treatment plant. And as you can see, that already includes 600,000 people, and the farthest point is already 8 hours away. And the question is, how different are these samples? We imagine that it would be good or it would be better to be closer to people's homes, since the information would be more fresh. But at the same time, we would expose ourselves to more noise and fluctuation in water usage by the population. This is some of the preliminary results. So as we can see in this slide, first, this is sort of the composition of the microbiome that we get at our manhole. And if you compare that to just regular human stool, as reported in the HMP, you can see that we are pretty similar, still dominated by bacteroidetes and firmicutes, though protobacteria have already started to also increase. And if you wait to sample at the treatment plant, the microbial community is already very different and quite dominated by protobacteria. And this makes a lot of sense because, you know, the two dominant groups in the gut microbiome are anaerobic. They don't like to be out there flowing in the water, while protobacteria are okay with that. And this is very exciting because this means basically that we can really use this data as a proxy for the microbiome of people. It's almost as good as going house by house to collect the poop. Which I'm sure they wouldn't appreciate. So the other thing that we didn't, so we're collaborating with a chemist at the Woods Hole Oceanographic Institute, Professor Lisko Jawinski, and we didn't have any hypothesis of how the chemical data would change. But so this was a very big, like, awesome surprise. And this is that not only we can find, you know, very common medications like ibuprofen or as commercially known Advil, but we can also see the glucuronide form of the medication. And this is incredible because, and if you wait, here you can see, you know, 3 samples we collected in our manhole, 3 samples we collected at the treatment plant, and by the time you get there, they are gone. And this is very consistent with other papers out there that look for glucuronides in their treatment plant samples and they don't see it. And this is because this glucuronide group is actually, you know, kind of easy to break off, and bacteria like to remove it to eat the glucuronide. But this is amazing because the fact that we see these molecules means that we can be 100% sure that somebody ingested ibuprofen and somebody peed out. Like there's no, you know, ambiguity that maybe somebody just decided to clean their medication cabinet and threw it down the toilet a bunch of medication. So I think I have just a couple of minutes more, but I will very quickly just mention that our very next step was to do a 24-hour sampling. That is, we collected water every hour for 24 contiguous hours. We had a team of about 40 people helping out, you know, collecting the water, bringing it back to the lab, processing right away. And we had to do this because we were wondering, okay, you know, we're much closer to people's homes. We have more data, that is awesome. But at what time of the day should we go and collect samples? Like, is it a time of the day in which people, you know, poo more? Or like we don't know, like we tried to make a poll in the lab and like, no, I'm in the morning, I'm in the afternoon, okay. We didn't have any consensus. Plus, we had the idea of, okay, maybe more people go in the morning, but is that going to be diluted by all the people who are also showering? So we had like all these questions, and then we were like, okay, maybe just let's go and let's do it. And basically, I mean, we were very happy that it actually really makes sense to what you expect. So here at the top, you can see the flow rate calculated as the average over 1 month at our manhole. And as you can see, the peak of the activity is 7 to 9 in the morning. So here, X is the time, Y is the flow rate, and you know you have peaks in the morning as well as at night. And and at night, you have quite a decrease of water usage, as you expect. And our data, as measured, our stool and urine data, which have been measured through mass spectrometry markers that we have found in mass spectrometry data, you can see they reflect the same trend. So, and you know, for example, stool in blue here goes basically to 0 at night, while urine doesn't. It goes down, but there's still some urine at night. And both of them were peaking in the morning as well. While during the rest of the day, they were kind of at the same level. So this was very comforting in the sense that, okay, you know, we're not just sampling noise. Like these samples actually still reflect what you expect from the population. And something also that was quite interesting is that we did 16S sequencing, some metagenomics, and we did the metabolomics from mass spec. And in the metagenomics, we were interested, okay, what are the plants that we're looking at? And it was great to see that, you know, the top hits were all like things people eat and are not quite digesting, I guess. So like maize, rice, wheat, and some weird things like pomegranate. Moreover, from the mass spectrometry data, now we have a method with about 30 common pharmaceuticals, including many of the most prescribed antibiotics in the US. That data is still being analyzed right now, but basically we have all of these are confirmed: caffeine, nicotine, artificial sweeteners, as well as very common painkillers. And we really see this as a platform to understand what people are consuming. Our very next experiment this summer is going to now expand to other locations and to start comparing. So we're going to have several sampling locations in Cambridge, in Boston, in Kuwait City, which is where our funding is coming from. And we're also trying to get things started here in Mexico, in the state of Veracruz, as well in Spain. These are just like, this is my last slide. Like a couple of weeks ago, actually, we visited Kuwait for the first time. As I was saying, the government of Kuwait is the sponsor of our project. So we traveled there for the first time to train people, but also to collect the very first set of samples over there. You can see them here. And I think, yeah, can you move? Yeah, I just really want to acknowledge all the people in the team, you know. Like, I'm very fortunate to be here talking to you and telling you about our work, but this is not like a one-person's job. And you can see we have this part of the work is mainly done by the ELM Lab, of which I'm part, the Sensible City Lab, which develops the robots as well as all the GIS data analysis of the maps, the Kujawinski Lab on the mass spectrometry data, and this wouldn't be possible without the collaborations also with Public Works, Public Health, and the wastewater treatment plant officers, faculty at Cambridge and Boston.
Thank you, Mariana. Let's move to the last session, the skin microbiome in Mexican patients with atopic dermatitis, given by María Teresa García Romero.
Hi, everyone. I'm very honored and excited to be here. Thank you for the invitation. So the subject of my talk will be the skin microbiome in Mexican patients with atopic dermatitis. Can I have the next slide? Or? Oh, okay, thank you. I am a dermatologist. I work at the Instituto Nacional de Pediatría. And this is a project that we're doing with Mariana Matus and the rest of the team at the ELM Lab at MIT. So the outline for this talk is to quickly review the existing evidence about the microbiome, both gastrointestinal and skin microbiome and atopic dermatitis, and to present the undergoing protocol that we're doing at both institutions, and also obtain feedback from the audience. So atopic dermatitis is an inflammatory skin disease. Its global prevalence in children is about 20%. However, it's one of the diseases that are more prevalent in developed countries than developing countries. And there's been a gradual increase in the last 2 decades that is not completely explainable. But one of the hypotheses that is strongest is a hypothesis that there's antigenic competence that, well, this means exposure to antigens and microorganisms early in life that sort of educate or regulate the immune system. And this is a complex interplay between the stimulation of the immune system and the development of tolerance. So I like this image because it's an image where we have 3 hypotheses that are very strong, and they have sort of evolved. So one is the hygiene hypothesis, where basically the more bacteria you are exposed to, the more tolerant your immune system will be. Yeah? That's the basis of the hypothesis. And that's sort of progressed to become the dual allergen exposure hypothesis. And what this means is, or the evidence shows, that the exposure to oral allergens or microbes basically educates the immune system through the GI tract, and this gives an environment where the immune system tends to be tolerant, versus the exposure to these allergens or microbes through the skin gives rise to a series of interactions, and what happens is the immune system becomes more prone to allergy or reactivity. And this is a big article that just came out earlier this year, and this shed some light on something that has for years been changing. Should children be exposed to allergens early in life? Eggs, peanuts, no, yes, no, yes. So basically what this study showed was that they enrolled 500 and something participants, that 550 had complete data. And what they did was, for 5 years, one group would not eat peanuts at all, and the other one would eat peanuts, basically, right? I mean, it's much more complex than that, but okay. And then they also stratified between patients who had skin prick testing for peanuts. The skin prick tells you that you are somewhat reactive to something. And then IgE, which is an immunoglobulin that tells you there is some kind of reactivity or allergy in your organism. Anyway, as you can see, the avoidance group is, the peanut allergy is much more prevalent in every group that had to avoid peanuts, compared to the group that ate peanuts or consumed peanuts, regardless of the skin prick test and the IgE results. So this has shed a lot of light on and basically supports the image that I showed you before. Then we have a lot of evidence about how, for example, skin colonization during a critical short period right after you are born is required to establish immune tolerance to commensal microbes. And what happens is that there is this high influx of Treg cells or lymphocytes into the skin, which creates an immune tolerance to commensals. So the cutaneous microbiome composition in neonatal life is crucial in shaping an adaptive immune response. And then there's evidence about how the delivery mode, vaginal versus cesarean section, and the feeding method, breast milk versus formula, are determinants of the intestinal microbiome in infants. In this study that was also published earlier this year, 102 babies were stratified and were followed up for the first few months of their life. And there was significant independent associations were found between the microbial community composition and the delivery mode and the feeding method. And the differences were larger between vaginally versus cesarean section delivery infants than the feeding groups. And another very interesting thing is that the microbiome of the babies who were mixed, who had mixed feeding, both formula and both breastfeeding, was more similar to the group that was exclusively formula fed. So, that gives you a lot of information to reflect on. Anyway, the gastrointestinal microbiome and atopic dermatitis. There's certain alterations in the gastrointestinal microbiome that promote an immune microenvironment that is prone to hyperreactivity and allergy, and there's certain degrees of evidence showing that, or at least suggesting that, maybe not proving, but suggesting. Specifically for atopic dermatitis, infants with the condition have decreased species of bifidobacteria in their feces. And this has given rise to the question, well, is this causal or just, a secondary finding? And one of the ways we can find that or get more information is by trying to modify the microbiome and then seeing if that has an effect. So this gives rise to the questions, are probiotic and or prebiotic supplements useful in modulating this microbiome and potentially decreasing the risks of these diseases? Probiotics, as you all know, are live microorganisms and provide benefits beyond nutrition. They've been used in a lot of inflammatory or reactive diseases under the hypothesis that they promote antigenic competence and then regulation and decrease susceptibility or intolerance to certain diseases. The previous studies using probiotics for atopic dermatitis have shown benefits, but there are some that have not been conclusive. Prebiotics are digestible inert fibers that give these probiotics a certain advantage to grow and to thrive. So they stimulate the growth of good bacteria. And there's a recent meta-analysis that showed prebiotics alone decrease the risk of developing atopic dermatitis in infants. And then there's the question of whether symbiotics, which is a combination of both, has a potentially greater effect because of synergistic action between probiotics and prebiotics. And we worked on this. We worked on this, a group and me, while we were getting our masters in public health. And we did this meta-analysis of the randomized clinical trials available using symbiotics, both for prevention and treatment of atopic dermatitis. And we found that using symbiotics provided a significant benefit in treating AD, especially in children that were older than 1 year of age, and symbiotics that had mixed strains of bacteria. We couldn't reach conclusions regarding prevention because there was a very small number of trials to analyze, and so we would need more studies to reach those conclusions. In terms of the skin microbiome and atopic dermatitis has for a long time been associated to Staph aureus, colonization and or infection, both as a factor to make the disease flare, to make the disease become recurrent, and also as a factor that promotes antibiotic use or bleach baths in order to decrease staph, and then patients ideally do, the idea is that they do better. So Heidi Kong and her group recently published a study. They wanted to study the relationship between the skin microbiota and disease, or AD, atopic dermatitis progression. And what they did was 16S RNA bacterial sequencing on serial samples of children with atopic dermatitis, and also a group of controls. And so here are some of the main results of their study. Here we have an image of what atopic dermatitis looks like. And so children were sampled at baseline, during a bad flare of the disease, and during the post-flare or after treatment for the disease. And what was found, I'm sorry, I think the image was cut off, I'm sorry. So what they found was that at flare, this is, the SCORAD is a scale or a score that tells you how bad the atopic dermatitis is. So during the flare, the diversity of microbial communities, which is cut off here, I'm sorry, but during the flare, the diversity was significantly decreased. Here's another way of, here's correlation between objective SCORAD and the Shannon Diversity Index. And here we have something also very interesting. So patients who had intermittent treatment, even if they were flaring up or clinically they weren't doing so well and they still had their dermatitis, the fact that they had intermittent treatment did not allow the Shannon Diversity Index to go down, as the patients who had no treatment had their diversity index go down while during their flare. So basically what their hypothesis is that the microbial diversity goes down during a flare, and one of the slides that didn't show, one of the parts of the image that didn't show was the fact that Staph aureus dramatically goes up while the rest of the community goes down during a flare. So here's that, the proportion of staph increases dramatically as the disease severity increases dramatically. So there's the question of whether staph is reacting to the changes in the skin, or staph is causing and causing the disease to be persistent. So our idea is to describe the skin microbiome in Mexican healthy patients and also Mexican patients with atopic dermatitis at different points of disease, just like Kong and her colleagues did, but we want to also do something different. So we want to compare the microbiome when patients are using different treatments. The standard treatment for atopic dermatitis is emollients, so keep the skin not dry, humid or humected, and using anti-inflammatories, which are usually topical steroids. So we want to compare this standard treatment to the same standard treatment plus intermittent bleach baths. And one of our ideas is, well, we are also in a country where people use antibiotics very frequently without a specific indication. This is a problem worldwide with serious public health implications. So if we could see that using bleach baths is better for atopic dermatitis, just like antibiotics are, we could potentially change the standard of care. And then we also want to correlate sequencing techniques, so 16S, and other metagenomic techniques, and also culture. We're going to include children 7 to 12 years of age who have no use of antibiotics in the last 3 months. I anticipate this is going to be a bit difficult because these children usually show up to our hospital and a lot of doctors have seen them and think they're infected, so they received multiple courses of antibiotics. We want to do 20 patients in each treatment arm and compare with 5 healthy controls. We want to sample multiple sites, especially in the first patients. We're going to sample more sites than the rest of the patients. And we want to sample during the flare and then once per month for up to 3 follow-up times from the flare visit, where patients will be actively treated. The idea is that they will get better, and then we will sort of leave them to their the natural course of their disease and then sample again. And that's it, and I'm available for questions. Thank you.
Okay, thank you, Maria Teresa. We are a little delay with the program. So we have time only for 3 questions. Okay.
So are you excluding children with mutation in the filaggrin gene? Because there's also an association with the filaggrin. That's this protein that puts together the collagen. So in this case, do you want to study these kind of people, or are you going to put them in a separate group, or you're not even looking at this gene?
Thank you. That's a very good question. The filaggrin gene mutation accounts for about 30 to 40% of patients with atopic dermatitis. Basically, what this defect does is that their skin is very dry and loses water. And that's just one of the factors in atopic dermatitis. Some patients don't have the mutation and they still have very dry skin and the immune alteration that gives rise to their disease. We are not planning on doing that, on stratifying patients based to their filaggrin mutation for now. That would be a very good question to do in the future, because ideally the patients who do not have hypothetically, the patients who don't have the filaggrin mutation have a stronger, so to speak, immune system alteration that is driving the disease. Thank you.
Okay, please, can you take your place? We have more time, a little more time. So,
hello. I'm sorry, I don't remember your name. The last one. Teresa, sorry. Very nice talk. I have a question about it. There isn't a relationship between malassezia and this kind of staphylococcus in atopic dermatitis in infant, or there is not any relationship between that? Because all of the words are bases with bacteria, but with malassezia and other yeasts that are common in skin, there's any relationship?
I see the audience has a very deep understanding about some of the skin conditions. So one manifestation of atopic dermatitis in infants is seborrheic dermatitis or cradle cap, where they have this thing of the entire skin microbiome, and they do bacterial, they do fungal, and they do viral. And in the skin, every site, every specific site has a specific microenvironment and a different microbiome. And malassezia is the 90-something%, well, I mean, it's a predominating fungus in our skin.
Okay, set.
Shirley, you showed in your samples that you were able to flip the proportion of human reads from 90% to 10%. Maybe I missed what the enrichment method was for you to be able to isolate the microbes away, but could you describe that again?
Okay. I mapped the clear sequence, the clear reads against the human genomes with SMALT, and then we obtained the sequences, the reads that not mapped with human. This is. Oh, okay. Experimentally. Yes, I have to filter the stool with pores. Yes, and we these filters was posed by 22 nanometers pores and 45 nanometers pores, and then we used chloroform to lysate the cells that could be in the samples, and then eliminate the DNA that the DNA that could be in the sample, and then we extract the DNA of viruses because they have a capsid. With this method, we could see these results.
Okay, thank you for the talks. I am totally amazed by so many of your data. And first for Jorge, I just have a couple of questions. You asked me just a couple of days ago, what did I think, how did I feel about picros and stuff like that, and your work is totally accurate, and I really love how you connect the diversity with the functional data and then perform this network reconstruction that is quite rare in the metagenomics study. So that's one is for you. And for you, now I realize why, for Mariana, now I realize how you were asking for translational things. No? So I'm totally amazed. But I don't know, are you thinking on taking this into like a genomic observatory analog or something like that, just to perform this regular data and release the methods so everyone else can follow you or something like that? I don't know if you can talk a little bit about it.
The method that I have been used, most of them are taken from the papers that the Human Microbiome Consortia or the MetaHit Consortia does. In particular, the ones that they use were more based on the ones that were they done for an article that that is based on the identification of complete genomes without the use of reference genomes. So with these methods, in theory, I can just recover even reference genomes without mapping, just of the correlation of the abundance of the gene. And well, yes, they are in the publication and they're quite easy to use to implement.
Okay, go ahead.
Yeah. Oh, okay. Yes, I think it's very important for me and the rest of the team that the methods we're developing and the data is available to everybody. And just to give you an example, the sampling robot that we have developed costs like less than $100 to build. If you compare that to existing commercial samplers that do worse than we do, and they cost like on the order of $15,000 each. So we really see this like as a transformative technology, and ideally we would want it to like extend to as many countries as possible. And also, you know, the data, as you say, one day can be a reference for anybody who wants to do research.
Okay, I'm sorry. So I have also one question for Jorge and one for Mariana. For Jorge, these patients you have, it's obvious that one of the problems is this frequent translocation of bacteria. My question is, have you done any microbiome on the blood of these patients?
No, we haven't performed this kind of analysis. We have detected the lipopolysaccharides into the blood by two markers. One is the CD14, and the other one is the BPI, which is a marker related to inflammation. And in this case, the inflammation given by the translocation of the lipopolysaccharides. And actually, we don't know if there are like complete bacteria, but we know that at least there are some pro-inflammatory components from the microbiota that now are in the bloodstream.
There is complete bacteria, and I guess there is a good variation of that in the blood. I mean, I just recall one recent paper on microbiome in blood, and it was not this kind of patients. So I guess in these patients you will see much more. So I would suggest do that. Maybe it's
a really good idea. Indeed, the fact is just show that actually exactly that is who is translocating, and if you are translocating, then are you triggering the immune response? And yes, I will love to. But I think, well, yes, I will. But we have to look for the serum and the blood from those patients.
And then for Mariana, this mapping you are doing in the cities, I guess you are considering the location where you are taking these samples. I mean, because it's not the same if you are working in a residential area, university, hospital, commercial centers, whatever. So the site is important. And then the other thing that I think it's important is the season of the year, because many infections, you have variations according to the season. So I guess you also want to consider these two factors, location and season, for your mapping.
Yes. So we have, generally speaking, two types of projects in the lab. So one of them that is looking at comparison of residential neighborhoods, and like the one in which I started and I will extend. And in that case, we're taking into account, we can start looking at questions of like ethnicity, age, socioeconomic status, and other variables that may be related to health. We also have other students who are looking more at particular sites, like hospitals, schools, maybe the airport or other important locations. So we don't know yet at this stage which one may be more useful or for what. So we're kind of tackling both. And regarding time, right now, once like we iterate a bit more in the robot, we hope that we can just start doing a lot of longitudinal sampling, like over winter. We have had for a long time the dream to be able to monitor the flu season in the States, or the norovirus seasons, which also take over like 2 or 3 times per year. So definitely like in the horizon.
For Mariana too, I want to thank you to everybody. It was a very nice lecture to all of you. And Mariana, have you also looked for antibiotics?
In the mass spec data right now, we have like 20 or 25 antibiotics, the most commonly prescribed in the US. I haven't seen the data yet, okay. But I just got an email yesterday that it was ready. And we also are going to start culturing the particular like bacteria to find what resistance they are carrying.
This is from Mariana too. So we're currently making a library of the 10 individuals from 17 animal species so that we can use those to map back to the sewage. So I'll give you those, and I'll show you the website they'll be at, because a lot of people dump their animal feces into the sewer. So we'd like to separate those out.
That's wonderful, and it will be super useful as well because we communicate a lot with like public works officers, and they very often they get like bad quality assessments in their outflows because it's based on CFU counts, and we're getting more and more requests to be able to untangle the source of the feces. So I think definitely would be super useful. Thank you.
I have a question for Jorge. Jorge, have you thought of using probiotics or symbiotics instead of prebiotics?
There are several studies that have used symbiotics in which they just give the short oligosaccharides and the bifidobacteria. And they haven't performed the analysis of the microbiota, but at least the systemic inflammation and the immune activation is decreased. So yes, this could be also an option. Yeah.
More questions?
A quick question about Staphylococcus aureus. Can you distinguish between aureus and epidermidis when you are sequencing the skin microbiome? Yes?
Yes. So in this one study by Kong and her team, Staph aureus was the predominant bacteria, but followed closely by Staph epidermidis, which in the past there was the thought or the belief that Staph epidermidis drove Staph aureus down, but Kong et al. Didn't find that. They go up simultaneously.
I also have a question for the skin project. Is there any difference between healthy people? We have leftists and we have right-handed people, right? So we usually use more one hand than other. So maybe there's some difference with the microbiota from one side to the other. Do you know something about that?
Yes. So the sampling sites are not the hand, but the usual sites where atopic dermatitis is more severe. And for some reason, that is the folds behind the knees. And then some parts usually don't get atopic dermatitis, like the anterior part of your forearm, right above your wrist. That theoretically wouldn't change if you're left-handed or right-handed, but that's an interesting point to make, of course.
Okay, let's stop here. You will have more opportunity to talk with the speakers. Let's thanks. And there will be some announcement from Esperanza Martinez.
No, from all of us. No, from all of us. Aquí nos quedamos paraditos. Well, this is the closing of the Congress. I hope you enjoyed it. And we want to acknowledge you for being here, our speakers that made a wonderful work. We also want to acknowledge our sponsors, UNAM, CCG, the Center for Genomic Science, and the American Society of Microbiology that also provided some funding. We want to thank people that have been involved in the organization of the flights and the hotels, especially Adriana Cortez, who did a wonderful job, and we should have her here. She's not here. We also want to thank the people that were involved in transmitting by video conference these presentations. So we have people that were working in Morelia, in Cuernavaca, and in Cuernavaca, Waldo Díaz, in Morelia, Ulises Olivares y Juan Carlos Franco, and here the transmitters were Antonio Ramírez, Ulises Olivares, Guadalupe Rojas, Fabián Martínez. They all did a wonderful job. And we also want to acknowledge the Colegio Nacional for receiving us yesterday. And we thank Emilia. Is she here? Emilia Sekeli, that made wonderful comments and contributions for planning the program. And we are here, the organizers, including Osvaldo. But I must thank especially Luis and Santiago Sandoval for making most of the hard work, because I gave the idea, he gave the money, he got the money, and they made the work. That's great. That's a good way to organize them. So now we have a surprise for you. We have some music upstairs, and there's people will sing, and also there will be some little snacks and bocadillos. Okay, welcome up.
It is
Please
upstairs.
go upstairs.
It's upstairs.